Hello. I hope everyone can hear me, and I want to welcome everyone to this wonderful event with Professor Forrest White. So before I speak to you today, before Professor White speaks, I'd like to just introduce myself and make a few announcements. My name is Sherry Marcus. I have a PhD from MIT in math from '93. And I am a member of the VP and board member of the MIT Club of New York. I'd like to make a few announcements just for the club. First, on Wednesday, September 16th, 2020, the MIT Club of New York will have an event for the women's community discussion, employment and labor in the COVID market. And 2 weeks later, on Wednesday, September 30th, 2020, we will have a virtual talk on looking ahead together on cybersecurity. So with respect to our illustrious speaker, Professor White, tonight, I'd like to just have a few introductions and background on Professor White and the Koch Institute for Integrative Cancer Research. MIT has a rich history of conducting cancer research beginning in 1974 with the creation of the Center for Cancer Research, the predecessor of the current Koch Institute for Integrative Cancer Research. The vision of the Koch Institute, which will celebrate its 10th anniversary next year, very exciting, is to fight cancer through the convergence of MIT's life scientists and the Institute's great strength of engineering. There are 27 faculty members at the Koch Institute, split between engineering and life scientists, as well as 29 extramural faculty members whose labs contribute to the mission of the Koch Institute. In total, there are almost 1,000 researchers united to fight against cancer. The convergence of life sciences and engineering at MIT accelerates the pace of cancer research. The Koch Institute boasts the highest rate of MIT co-authorship in its publications, surpassing all other departments. Even though patients are not treated at the Koch Institute has a number of signature partnerships with hospitals and industry to get their work into the clinic. They are currently engaged in 25 clinical trials, including one of which you will hear from Professor White this evening. With that, I'm now pleased to introduce our speaker. Professor White is the Ned C. and Janet Bemis Rice Professor of Biological Engineering at MIT, a member of the Koch Institute for Integrative Cancer Research, and a member of the MIT Center for Precision Cancer Medicine. He earned his bachelor's degree in chemistry from Framingham State College in 1993 and a doctorate in analytical chemistry from Florida State University in 1997. Following postdoc research in bioanalytical chemistry in the laboratory of Donald Hunt at the University of Virginia, he joined MDS Proteonics as a research scientist working up to a group leader. He joined MIT in 2003 as an assistant professor and was awarded the Mitsui Career Development Professorship in 2005. In 2010, he received the Ruth and Joel Spira Award for Excellence in Teaching. The goal of the White Lab is to define novel therapeutic strategies using combinations of cancer therapies or combinations of chemotherapies and targeted immunotherapies, which you will learn more about tonight. Before I turn it over to Professor White, please note that you may submit questions at any time during this Q&A using the Q&A feature at the bottom of the screen, and we will do our best to get as many questions answered. Thank you again, and we will now leave it to Professor White.
Thanks, and thank you for the introduction, and thank you for attending today. All right, I'm going to share my screen. All right, so what I'd like to talk to you about today is our method, So what I'd like to talk to you about today is our approach to use systems biology to uncover new therapeutic strategies for incurable cancers. And specifically, I'll be talking about triple-negative breast cancer today. Cancer and other human diseases are fundamentally driven by dysregulated cell behavior. In cancer in particular, this dysregulated cell behavior comes in the form of aberrant proliferation, aberrant migration, or invasion. as well as pro-survival where other cells might be dying. Many of these cell behaviors are controlled by the pathways shown on the screen here, as previously highlighted by a nice review from Doug Hanahan and Bob Weinberg in 2000. Although this network that's shown here looks pretty complex, it turns out that it's actually just a fraction of the signaling networks that actually control these cell behaviors and are dysregulated in cancers. In order to understand and treat cancer as well as many other human diseases, we really have to understand the complex regulation among these different biological networks, because understanding that complex regulation allows us to start understanding how we can treat these diseases and basically stop this dysregulated cell behavior. Many people look at these diseases through a variety of different lenses. We can start with sort of the central dogma of biology, which is that DNA, which is sort of the blueprint of the cell, gets transcribed to RNA, and then this RNA gets translated into proteins. And then when these proteins are going to be activated, typically they get decorated with different post-translational modifications or chemical moieties that are added to the surface of these different proteins. Many diseases, including cancer, can be driven by genetic mutations. These genetic mutations get converted into RNA mutations and then into mutant proteins. And typically these mutant proteins have either loss of function, which is actually very challenging to treat, or gain of function, or even altered activation state. In the gain of function case and in the altered activation state case, we typically have a change in the post-translational modifications of these proteins that we can then identify and quantify if we want to understand what's regulating the protein activation. To measure these different mutations or to measure these different properties of cancer and tumor cells, many different techniques have been used. Cancer genomics has been used extensively over the last 10 years or so to identify the mutations present in different cancer types. Many companies, as well as many researchers, have used transcriptional profiling to come up with signatures of different cancer types and different cancer subtypes. We've focused on proteomics, which is the study of proteins, and our specific focus is on functional proteomics. This is the identification and quantification of protein post-translational modifications that are regulating protein activities. The questions we aim to address with our approach are really to try and change clinical diagnosis by directly identifying activated pathways regulating critical processes in cancer, as well as using functional proteomics to identify therapeutic combinations and therapeutic strategies to overcome resistance and make current therapies better. What I'll talk about today is actually mostly this first question about clinical diagnosis and trying to identify activated pathways in human tumors that we can then target clinically with drugs. Our main focus is on protein phosphorylation. This is a post-translational modification where protein, a kinase, basically takes ATP and converts it to ADP, and then takes the phosphate from that ATP and puts it onto another protein. This protein then typically changes activity, although it could change as well interaction partners and shape and a variety of other behaviors. But typically, what we'll be studying is a change in activity of these proteins. Interestingly, when we look at kinases, most of the oncogenic drivers that have been identified are dysregulated kinases. And accordingly, most of the targeted therapeutics for cancer actually target these activated dysregulated kinases. And so by monitoring protein phosphorylation, not only can we monitor the kinases themselves, but we can monitor their activity in the cells and identify potential therapeutic targets this way. Among the phosphorylated proteins that we could study, there's phosphoserine, which is basically a phosphorylation of the serine amino acid, and that constitutes 90% of the phosphorylated residues. Phosphothreonine constitutes about 10% of the phosphorylated residues. And the remaining residues, which is a very small fraction of the total phosphorylated protein in the cell, are phosphotyrosine residues. And these are about 0.1 to 1% of all phosphorylated residues on proteins. Phosphotyrosine is not only very low abundance, but it's also very highly regulated. And in fact, tyrosine kinases make up the largest class of targeted therapeutics for cancer. And so not only are these phosphorylation sites very rare and very tightly controlled, but they're also very important in regulating proliferation, migration, and invasion of cancer. So these are what we'll be studying. If we look at one of the very well-known pathways in cancer, this is the epidermal growth factor pathway. And between epidermal growth factor, HER2 and HER3, these are the proteins that are often dysregulated in breast cancer, as well as many other types of cancer. Every place where there's a red circle, we see a tyrosine phosphorylation site on these proteins that can regulate its activity and therefore regulate the signal transduction that's occurring from dysregulated EGFR or EGFR family members. So by monitoring tyrosine phosphorylation on this network, we can identify the dysregulation within this network and identify potential therapeutic targets for tumors driven by EGFR or a variety of other different drivers. Okay, so my main focus for today's talk is going to be on triple negative breast cancer. And I'll just give you a few statistics here without belaboring the point too much. There's an estimated 270,000 cases of breast cancer in 2020. The good news is most of those patients are going to survive for more than 5 years. In fact, approximately 90% of those patients will survive for more than 5 years. However, there's a small number of patients, well, not a small number of total patients, but a small percentage of breast cancer patients, 15% or so, that are classified as triple negative because they lack HER2, they lack estrogen receptor, and they lack the progesterone receptor. In other words, they're HER2 negative, ER negative, and PR negative. For triple negative breast cancer patients, the frontline therapy is chemotherapy and typically paclitaxel plus a platinum agent, often carboplatin. Patients that receive this chemotherapy, about 40% of those triple negative patients are going to have a complete response to therapy and have extended life expectancy. The other 60% of those triple negative breast cancer patients, they have an incomplete response, and this is associated with typical poor survival. Unfortunately, for those patients, the therapeutic options are minimally effective. Currently, they're given checkpoint inhibitors, which have actual minimal effect for triple negative breast cancer patients. And the other therapeutic options that have been tested have not necessarily been more efficacious. So in chatting with the clinicians who work on this disease, there's really a lack of good options for these chemotherapy-resistant triple-negative breast cancer patients. So a couple of years ago, we got involved in triple-negative breast cancer and specifically with the breast cancer team at the Mayo Clinic in Rochester, Minnesota, shown here at the bottom. Matt Getz, Judy Bowie, and Li Wei Wang are excellent researchers and clinicians, and they started what's known as the Beauty clinical trial. And this Beauty clinical trial basically took 140 women with invasive breast cancer, and when they were diagnosed, they took a biopsy from those patients, and from that biopsy, they were able to take parts of that biopsy, inject it into mice as a xenograft tumor, and then grow that tumor in mice and propagate that tumor in mice so that you could not only characterize the tumor better, but test different therapies on that tumor. After diagnosis, the tumors were split into either HER2 positive or HER2 negative tumors. And the HER2 negative tumors were given either paclitaxel or for the ER positive patients, or paclitaxel and carboplatin for the triple negative patients. After a couple of rounds of chemotherapy, the tumors were resected, and that tumor tissue was then again, parts of that tumor tissue were again injected into mice as patient-derived xenografts that again can serve as avatars for the patient to see which drugs the patient might respond to based on what the mouse tumors are responding to. And then these patients were put on 5-year observation. And we've been working on a variety of different mouse xenograft tumors from this beauty trial in collaboration with the team at the Mayo Clinic. So the triple negative breast cancer tumors retain the chemotherapy response of the patients. And that's shown here at the top. For instance, we have a patient where you can see the tumor before paclitaxel, and then after paclitaxel, the tumor is gone. This was a complete responder to the treatment. The patient-derived xenograft tumor of that same, so the mouse tumor effectively of that same tumor, has a very similar response where treatment with paclitaxel dramatically shrinks the tumor volume compared to vehicle. At the bottom, we have another patient that was a clinical non-responder. You can see that that tumor is still there after paclitaxel. And again, the patient-derived xenograft tumor, the mouse tumor of this patient tumor, basically shows the same behavior where we get a minimal response to paclitaxel in the animal. And so these PDX tumors appear to faithfully reconstitute the patient response to paclitaxel. And these mouse tumors are what we've been studying in order to identify potential therapeutic targets for these chemotherapy-resistant patients. So what we've done with the Mayo Clinic is to start looking at some of these different patient-derived xenograft or PDX tumors. And for each one of these, the patient tumor cells are injected into the mammary fat pad. The tumors are grown to a couple 100 millimeters cubed, and then resected and frozen and shipped to us where we do our quantitative functional proteomic analysis. The details of that are shown below here, but I'm not going to go through the workflow except to say that we basically pull out our tyrosine phosphorylated peptides and then identify and quantify those tyrosine phosphorylated peptides so we can read out the activated signaling networks in these different patient tumors. As stated here, we've analyzed about 60 of these triple negative breast cancer PDX tumors, including many chemotherapy resistant and chemotherapy sensitive tumors. So we start looking through that data. We see data like what's shown here, where on the left side of the screen, we have a chemotherapy sensitive tumor. And this is the signaling network that we've identified from that chemotherapy sensitive tumor. On the right side of the screen, we have a chemotherapy resistant tumor. And again, the signaling network, which is actually quite complex in this case, there's a fraction of it shown here. The signaling network from this chemotherapy resistant tumor is shown here. So we can take the chemotherapy resistant tumors and the sensitive tumors and start comparing them against each other. And what we identified from this is what's known as a Src-family kinase network that was enriched in the chemotherapy resistant tumors compared to the sensitive tumors. And this Src-family kinase network is really dominated by these 3 proteins in the middle, FIN, Src, and YES. These are 3 of the classical, what are known as Src-family kinases. Src was the first oncogene that was identified as a tumor driver. And it's been a classic sort of cancer target, although clinical trials with Src inhibitors have been not very effective when the Src inhibitor has been given as a monotherapy for most solid tumors. But what we see in our data is basically enrichment of this Src-family kinase signaling network in the chemotherapy-resistant tumors. And on the right-hand side of the screen is a drug called dasatinib or Sprycel, which is a Src-family kinase inhibitor. And this drug is clinically approved for a particular type of leukemia, but so far has not done well in treating solid tumors. So we wanted to see whether dasatinib, which is really the right drug to attack this network, is effective or would be effective at treating these triple negative breast cancer tumors. So we picked out a tumor that was a Src-family kinase high signature, and that's shown here. And basically the drug treatment with dasatinib or paclitaxel or the combination thereof was started when the tumor was fairly small and about 50 millimeters cubed or so. And then the drug treatment was stopped at day 21. And then the tumor size was then monitored all the way out to day 76. And what you can see from this data is that the mice are irresponsive to or unresponsive to paclitaxel. As expected, this is a chemotherapy-resistant triple-negative breast cancer tumor. But however, they're actually very nicely responsive to dasatinib. In fact, dasatinib basically stops tumor growth, and that tumor growth is stopped for another 30 or so days after treatment was stopped, at which point, by this point, the drug is completely gone from the system and the tumor starts to basically grow again. But still, when the mouse was on drug, we basically get a complete stoppage of tumor growth. So this worked really well for this one tumor with the SFK high signature. As we look at other mouse tumors, what we see is that for another tumor with SFK high signature, as we start the drug, again, the tumor basically stops growing when we have dasatinib or dasatinib plus paclitaxel, whereas the vehicle basically continues to grow throughout this time. For one of the tumors that had a low SFK signature, we predicted that it would not be as responsive to the drug. And indeed, what we saw is that the mouse was, again, not responsive to paclitaxel, but basically was somewhat responsive to dasatinib, but not as responsive as the SFK high signature animals, which is basically what we predicted. And so we did a couple more of these and showed that our predictive capability was actually quite good for identifying mice that had, or these mouse tumors, mouse patient-derived xenograft tumors that had high SFK signature would tend to respond quite well to the tumor, to the dasatinib treatment. Okay, and so the data actually looked very promising, and the clinicians at the Mayo Clinic were actually very excited about this, but some critical questions remained at the end of this part of the study. The first one being, how physiologically relevant are these PDX tumors? They're human tumors that are grown in a mouse, and so are the signaling networks that we're seeing in these tumors actually representative of human tumors? The bigger question, and the one that was really vexing us, is why did dasatinib fail in the clinic? And dasatinib, this drug that we showed worked quite well on the SFK high chemotherapy-resistant triple-negative breast cancer tumors, was already tried for triple-negative breast cancer in a clinical trial and didn't do well. And so this raised the question of how common is this SFK high signature in triple-negative breast cancer patients? We've been looking at the data, and one of the questions, or the big question that emerged is, from the clinical trial, this was an all-comer clinical trial, is it that only a small percentage of those patients were SFK high and therefore the drug didn't work? Or is it that a lot of the patients were SFK high and yet the drug still isn't effective in the human case? And so we really need to answer that question, because there's a chance to actually resurrect this drug and save this drug and potentially treat a fraction of the triple-negative breast cancer patients. To answer these questions, however, we really need to be able to look at human tumors. And one of the challenging things at this point in the study was that we had never performed our phosphotyrosine analysis on what's known as FFPE, our formalin-fixed paraffin-embedded tissues from human patients. And typically when human patients get resected in the clinic, that tumor or parts of that tumor are preserved by fixing them in formalin and then embedding them in paraffin, or generating these FFPE tissues. So we wanted to be able to perform our phosphotyrosine analysis directly on these FFPE tissues from human patients. So one of my graduate students, Ishwar Kohale, basically developed a new approach that allows us now to look at phosphotyrosine signaling in FFPE tissue sections. We did a proof of concept on glioblastoma, patient-derived xenograft FFPE blocks, and with Jan Sarkaria, another clinician at the Mayo Clinic. Glioblastoma is sort of a classic, it's a devastating and difficult to treat form of brain cancer. And we've been working on glioblastoma with Jan Sarkaria for a while. But what we showed in this sort of proof of concept study is that we could take the FFPE blocks take a couple of 10 micron sections from those blocks, and then process it through this new method that Ishwar developed to be able to analyze and quantify tyrosine phosphorylation from these FFPE tissues. The data briefly is shown here, and the important part is shown on the right-hand side at the bottom of the screen here, that basically shows we can quantify kinases and the activation site on many kinases in these FFPE tissues from these different, from this PDX tumor. We took this approach and applied it to triple negative breast cancer samples from the clinic at the Mayo Clinic, again in collaboration with Matt Getz and the Beauty team. And they identified 10 triple negative breast cancer patients who, upon resection, had their tumor, parts of their tumor frozen in liquid nitrogen, or parts of their tumor fixed with FFPE. So we took both parts of those tumors, or both parts of that tumor, and analyzed them separately. And the nice thing about this data is that it showed that the frozen tumors, which are sort of the gold standard in the field, are closely matched by the fixed tumors. So our data analysis shows that the signaling networks from these FFPE tumors are closely aligned with the signaling networks from the frozen tumors. More importantly, when we start looking at these FFPE tumors, we can start quantifying the top 10 oncogenes or so from these different tumors. And what we can see is that, for instance, patient 1 has a very high EGFR phosphorylation and has very high EGFR activity in this tumor. There's actually good inhibitors against EGFR, and it's likely that this patient might actually be responsive to one of these inhibitors. On the other hand, patient 2 has some EGFR phosphorylation, but actually is driven mostly by c-Met, which is another receptor. And again, there's actually a couple of good therapeutic options for inhibiting c-Met. As we start looking through these different patients, one of the interesting things that was revealed is that only one of these 10 patients had what we would characterize as a Src-family kinase high signature. So out of these 10 patients that we analyzed, only one of them we predict would be responsive to dasatinib. Of course, 10 patients is a very small number, and it's hard to extract good statistics from this small number of patients. But to address this, we're getting an additional 400 or so FFPE tumor tissue sections sent to us for analysis so that we can actually start characterizing how many patients actually have this SFK high signature, especially in chemotherapy-resistant triple-negative breast cancer. Once we're through that analysis, we can start trying to figure out whether it's worth trying to resurrect dasatinib and see whether this might actually be a good clinical strategy for some of these patients going forward. So in the last part of the talk here, one of the challenging things that's facing us is trying to identify these SFK high patients for treatment stratification. What are the options to really develop a biomarker signature, and especially a biomarker signature that we could use in the clinic? And equally importantly, can we make our phosphoproteomic strategy that we use as sort of an art form in my lab, can we make this more turnkey and really accessible for clinical samples in hospital core labs, where patient tissues can then be analyzed in the hospital core lab to identify the activated networks and therefore stratify those patients onto different therapies. So for the first option to develop a biomarker signature, we turned to RNA sequencing and did transcriptional profiling of 64 of these PDX tumors. And what we see is that we have basically multiple copies of different PDX tumors from 4 different patients. And what we see is that those tumors basically are characterized by the patient from which they were derived. If we dig in deeper to the data, what we see is that unfortunately, clustering of the transcriptional data has not revealed a Src-family kinase high or Src-family kinase low, or any sort of Src-family kinase dependent signature. And so this was a little bit frustrating because we were hoping that we could use the transcript data that's already there in abundance in the clinic to identify which patients could be Src-family kinase high. So it turns out that hasn't panned out very well. And so we've turned to trying to develop now a better approach for phosphotyrosine analysis that could be used in hospital cores for clinical samples. And we've done this in collaboration with Aaron Gadjadar, senior marketing specialist at Thermo Fisher Scientific. They're the people that actually make some of the mass spectrometers that we use. And in collaboration with Thermo, we've now developed this technique that's shown here. I won't go through all the gory details here, but the important thing to know is that this technique basically allows us to go in and using commercial instruments and commercial columns and commercial setup, we can now basically identify and quantify tyrosine phosphorylated peptides from clinical samples and do it actually quite quickly. And so our goal now is to move this out so that hospital core labs can start doing the same analysis and making some of these same predictions that we can make. So some of the data from that is shown here, and I'm just going to skip through this quickly in the interest of time. But basically, this phosphotyrosine analysis provides a direct readout not only of the signaling networks within the tumors, but also the signaling networks of the immune cells around the tumors, which will inform on some of the immunotherapies that are currently being used in the clinic. In summary, what I've talked about today is that we've identified this SFK high subset of chemotherapy-resistant triple-negative tumors and shown that they actually respond well to dasatinib, potentially in combination with chemotherapy, but even being treated with dasatinib by itself. We've shown that we can use functional phosphoproteomics and tyrosine phosphoproteomics to identify these patients. Sorry. And lastly, this combination of this new method, short quant phosphotyrosine, plus the FFPE tissue analysis approach that we've developed, can identify other potential therapeutic targets in triple-negative breast cancer, as well as other cancers. We're now extending this approach to glioblastoma, as well as pediatric brain cancer, colorectal tumors, lung cancer, and pancreatic cancer. And the response from the clinicians has actually been quite strong because they see that we can start identifying these activated networks directly rather than trying to use cancer genomics or transcriptional profiling as a proxy for network activation. So lastly, and most importantly, are the people who have done all the work. Most of the research I talked about today was done by Ishwar Kohale, a graduate student in my lab. The short quant phosphotyrosine work was done by Lauren Stauffer and Cameron Flower, two other graduate students in my lab, in collaboration with Aaron Gadjadar at Thermo. The rest of the White Lab has actually been incredibly important in contributing to this work and enabling the work. And the breast cancer collaboration has been done with Matt Getz, Judy Bowie, and Li Wei Wong at the Mayo Clinic. And the glioblastoma work has been done with Jan Sarkar at the Mayo Clinic. We can't do this work without support, and we've had tremendous support from the Koch Institute, as well as the MIT Center for Precision Cancer Medicine, as well as great support from NIH and the National Cancer Institute. And lastly, here's a picture of my group social distancing with masks on at a recent celebration. And so we all have to adapt to the coronavirus, and this is one way of us adapting to the coronavirus. So thank you very much for your attention, and I'd love to take questions.
Thank you. So first question, thank you for your talk, Professor White. For cancers beyond the blood-brain barrier, how does drug delivery work for what you have spoken about? Do these chemicals pass the barrier?
That is a fantastic question. And we've been working, we actually have a center that was funded by the National Cancer Institute to look at the effects of the blood-brain barrier and heterogeneous delivery of drug into brain tumors. And it turns out that that drug delivery is absolutely critical. And so we've been trying to identify therapies that have high brain penetration and then characterize their behavior on different tumors.
Mm-hmm. Thank you. Next question is on targeted immunotherapy. Do you have any comments on use of stem cells to cross the blood-brain barrier to carry therapeutic drugs or prodrugs to glioblastoma and/or metastatic breast cancers to brain?
Wow. That's another great question. Yeah, so both stem cells as well as activated T cells cross the blood-brain barrier and can be targeted to human tumors. That's a really interesting idea, and people have been starting to use activated T cells for some of this, as with backpacks, for instance, that carry chemotherapies to the tumor. I haven't heard of people using stem cells that would home to the tumor for that, but I think the concept is still the same. There's a lot of work, I think, to be done to do that well, and especially to get enough payload on those cells and get them to the right place at the right time.
Thank you. So the next question is, how common do you think this phenomena is in clinical trials, i.e. the drug was written off as unsuccessful, but in truth it worked well, but just in a small group of people?
This is my uneducated guess or uneducated estimate on this. I actually think it's very common. I think that from what we've seen in a variety of different tumors now, as we start looking at human patients, the heterogeneity among different patients is very high, and trials that take all comers are almost destined to fail, because without some improved stratification, many of those patients just do not have the right activated networks that would be susceptible to those drugs. So my goal, and part of the goal of looking at these larger number of triple negative breast cancer patients, is to not only identify which ones are Src-family kinase high, but to identify other potential therapeutic targets, and more importantly, identify signatures of those so that we can accurately stratify those patients.
Thank you. Next question is, how has the pandemic affected your work in these clinical trials?
Yeah, these are all great questions. So I'll stop saying that's a great question, but they're all good. It's been really challenging. So the pandemic shut down our lab for, I believe it was 3.5 or 4 months, where we were not doing any research. We were able to get some papers wrapped up and sent out during that time, but we really were unable to sort of push some of these projects forward. It also sort of affected the researchers at the Mayo Clinic and the clinicians at the Mayo Clinic quite a bit. And so they basically had to delay, especially on the glioblastoma side, delay some of the work that we've been doing with them to move a couple of drugs into phase 0 or phase 1 clinical trials. So it's actually had a pretty profound effect. We're starting to rebound from that pretty nicely now. And, the lab is up and running, and we're generating data and hopefully moving this project and hopefully clinical trial forward soon.
That's wonderful. How close are we to actually implementing a mass spec analysis of patient samples at a pace fast enough to change the course of treatment for that patient? What would be some of its limitations?
Yeah, that is exactly what we're working on. And that's exactly why we have this collaboration with Thermo to try and enable sort of higher speed analysis of some of these samples. With this approach, we can do on the order of tens to hundreds of tumors per week. And so we can easily turn around tumor samples in a timely fashion that would get us answers for some of these patients within days. So after being resected or after biopsies have been generated. So moving this technology now, our goal is to move this technology out to the hospital core labs so that the samples don't have to be sent to MIT because we would get swamped pretty quickly with that. But I think if we can transfer this technology now to hospital core labs, we can easily implement this. So there's some approval steps that would have to be taken, and that's something that we're going to have to do in the near future.
Okay, that's great. So I don't see any more questions on the Q&A. If it's okay, if anyone of our attendees has a question they would just like to ask directly to Professor White, we can open it up. Got another question. In addition to triple negative breast cancer and glioblastoma, are there any other cancer types to which you hope to adapt this technology?
Yeah, so we've been working in collaboration with research at the Dana-Farber Cancer Institute on pediatric glioma, a particular nasty form of that's called DIPG, and trying to identify, also with researchers at the University of Pittsburgh Medical Center, signaling networks in DIPG, as well as why this first-in-human trial did not go well for the patient on which it was tried. So we've been working on DIPG. We're starting to work on pancreatic cancer, lung cancer. We've started working on colorectal cancer and are launching a bigger project in that area. So I'm very excited about tackling some of these, like the title indicated, incurable diseases. In many of these cases, patient survival is less than a year after diagnosis, and there's just no good therapeutic options. And so I would love to change the way that these patients are treated.
Thank you. Any more questions from the audience? Yes. In one of your charts, you showed results for up to 29 days. Do you have data for longer periods?
Yeah, that's again another good question. So the clinicians at the Mayo Clinic basically took one of the tumors out to 76 days, the other tumor out to 29 days, and then basically had the other ones out at 21 days or so. They basically were stopping the treatment when they stopped the drug because we had already shown sort of what the drug was doing. What they didn't do, which would have been actually great, would be to keep the drug on for longer than 21 days. Unfortunately, the cost of the drug is not cheap, and giving it to animals ends up costing quite a bit of money, and there wasn't that much money to support this project at that point. But I do think that's something that we will have to do as we start moving forward, especially if we identify that there's a portion of patients that might actually be responsive to this drug.
Yeah. Well, Professor White, thank you so much for this talk. It was amazing. We have another question. Keep on coming in a little bit. You spoke, and we have some time. You spoke of 10 human tissue samples, and you spoke of 400 more samples. What is the total number of candidate samples?
So we're basically going to have about 410 candidate samples coming in that are triple negative breast cancer patient tumor specimens from the Mayo Clinic. So from that data, we hope to be able to identify the frequency of the SFK high signature, as well as frequency of other potential targets that might emerge from that analysis.
Okay. Okay. Is there support for this project right now?
There is some support. Based on it, we recently got another grant from the NCI to address new therapeutic targets in triple negative breast cancer in collaboration with the Hebrew University in Israel, as it turns out.
That's fantastic. That's fantastic. Okay, well, going once, going twice. Thank you so much, Professor White, for this talk. Again, it was fantastic. We really appreciate your time, doing these Zoom talks. It's very different for the club, but we all enjoyed it very much. And thank you again.
Yeah, well, thank you very much for moderating today and for the invitation to give this talk today to the club. And thank you all for attending and for the excellent questions. That was great.
Thank you. We appreciate it.
Excellent.
Okay.
All right.
Bye-bye,
Bye.
Owen. Thank you, guys. Bye-bye.